You must have heard about the terms like chromatin, chromatid, nucleosome, sister chromatids, chromosome. So you must be wondering what is the difference between all these things. Hello, I'm Dr. Kamya Tikku, and in today's video, we are going to depict the differences between, especially between chromatin and chromatid, which are often confusing terms. So always remember that whenever a linear DNA, which is the form of DNA present in eukaryotic organisms, whenever linear DNA associates with histones, because DNA being negatively charged and histones being positively charged, they form bonds with each other. And this is also a requirement of the cell so that we can package a linear long DNA inside a small nucleus. So, for packaging and organization and managing the DNA inside the nucleus, this long DNA, which is negatively charged, is joined with positively charged histone proteins. Now, the next step is DNA and histone proteins together, they will form a loosely formed complex called nucleosome. When this nucleosome further condenses, it forms chromatin. So, chromatin is the loosest and least organized form of a cell's genetic material. It floats freely around the defined envelope of the nucleus. It is the complex of DNA and protein found in the eukaryotic nucleus which packages chromosomes. And the structure of chromatin varies significantly. You can see during interphase, two types of chromatin can be found, like euchromatin and heterochromatin. Euchromatin means true chromatin. Heterochromatin means that portion of the chromatin area, that is DNA and protein complex, which is still not discovered, or if you try to check it under the microscope, it will remain dark. Whereas this area, because it is not that tightly arranged, so this is illuminated, and we can see this area under the microscope. So once the chromatin structure is formed, this chromatin structure further condenses during cell division to form chromosomes. Each chromosome has a chromatid. These are called sister chromatids to each other. So a chromatid is one of the two identical copies of DNA making up a chromosome that are joined at the centromere. So this portion is called centromere, to which two sister chromatids are combined. During cell division, the chromatids separate and move to opposite end of the chromosome, ensuring that each daughter cell receives an identical copy of the genetic information. So thus, now I hope you must be able to understand the difference between nucleosome, chromosome, chromatid, and chromatin. This is centromere, which is holding two sister chromatids together. What are chromatids? They are two halves of a chromosome. What is chromosome? It is a condensed form of chromatin. What is chromatin? It is loosely formed arrangement of protein and DNA. What is nucleosome? It is more loosely formed arrangement of DNA and protein. And that's all for the video.